Try to, like, just for the days you have classes with me, try to arrange that laptop, okay?
Okay, ma'am.
All right. Yes, everyone, switch on your videos. Come on quickly. Quickly, without me having to ask you 10,000 times. Also, did you people have any difficulties from the previous lecture? How hot is it at your place, students? On a scale of 1 to 10, how hot is it?
7.
7.
10.
10. Who said 10? Fez. It's 10 at your side.
Yes, ma'am.
Oh, God.
Yes, ma'am. It's very hot.
Okay. And what about Jobita?
8.
8. Zanab?
8.
8. I feel like it's 12. Like it's beyond this scale. It's so hot. It's like so unbearably hot. Okay, everyone, switch on your videos. Quick, quick. Are you people doing the homework and submitting the homework on time? Hmm? The question is to everyone. Hmm? The silence, sometimes I feel it's very awkward to have this silence. Okay, students, let's just do a quick revision. Then we'll start. Faz, even I can't see your video. Yours is also not visible. Do something about it. Hashid, switch on your video. Sabiha. Sabiha, are you back or are you still in India?
Ma'am, I'm still in India.
You're still in India. Would you try switching on your video today?
Ma'am, I'm in the Gao and like I already don't have like really good data.
Okay. Where is your village?
What, ma'am?
Where is your village?
In Andhra Pradesh, ma'am.
Okay, okay. So you here for your summer vacations?
Yes, ma'am.
Okay. And all of your summer vacations have started, students?
No,
No,
ma'am.
ma'am.
No.
No.
Oh. Okay, let's just revise what we have discussed. Now we have started the chapter cell. Now cell is the unit of life. Now cell is the structural and functional unit of life. All the living organisms are made up of cells. Depending upon the number of cells, the organisms can be unicellular or they can be multicellular. Now unicellular organisms are those that are made up of just one cell, such as amoeba, diatoms, bacteria. Multicellular organisms are those that are made up of more than one type of cells, such as plants and animals. Now, anything less than a cell is not considered as living. So that is non-living. So this is the reason why we call cell as the structural and functional unit of life. Now, one cell is capable of existing independently and it is capable of performing all the essential necessary functions that are responsible for, like you can say, continuing of life. Now, who was the person who discovered the cell first time? It was Robert Hooke. He discovered the first cell, but the cell that he discovered was a dead cell. Then Anton von Leeuwenhoek, he was the first person to discover the first living cell. Then more and more studies started to happen around the cell and a cell theory was formulated. Now the cell theory, which was formulated by Schleiden and Swan, it said initially what happens is that Matthias Schleiden, who was a German botanist in the year 1838, he studied plants and observed the plants and he said that plants are made up of different kinds of cells. Then came Theodore Swan, who was a British zoologist and he studied animals. So both of them said that plants and animals are made up of cells and products of cells. But they did not explain the origin. Origin meaning where are the cells coming from. So then came another scientist whose name was Rudolf Virchow. Now he explained that all the cells divide and form the new cells, meaning the cells come from the cells that have already been existing. So the cell comes from pre-existing cells. And this phenomena is known as ominous cellular e-cellular. Now, finally, the cell theory was created. And these are the three most important points about the cell theory, that all living organisms are made up of cells and products of cells. All cells come from pre-existing cells. And then activities of an organism are the outcome of sum total of activities and interactions of the cell. Finally, let's see where is it applicable. So the cell theory, this theory is universal. It's applicable to every single organism except viruses. Viruses are an exception to the cell theory. Now, why they are an exception? Because they are dead outside the body of the host and inside the body they are living. So they are not actually truly living organisms. Then talking about the components of the cell. So cell has three basic components such as the cell membrane, the cytoplasm and the cell organelles. Now, the organisms are of two types. Now what, like prokaryotes and eukaryotes. Now who can tell me what is the basis of difference in between them? What is the difference between prokaryote and eukaryote?
Prokaryotes, they have a membrane-bound nucleus and membrane-bound organelles, and eukaryotes have a prokaryotes do not have a membrane-bound nucleus, and eukaryotes have a well-defined membrane and a
Very,
membrane-bound nucleus.
very good. So exactly like how Jesslyn said, prokaryotes are all those organisms that do not have a membrane-bound nucleus and they do not have a membrane-bound cell organ. Then eukaryotic are the ones that do have. Now, prokaryotes are the most primitive organisms, and eukaryotes are both advanced ones. The cell size of prokaryotes is smaller, and the cell size of the eukaryotic organisms are larger. Ribosomes are 70S type for prokaryotes, and they are of 80S type for eukaryotes. Centrioles are absent in the case of prokaryotes, and centrioles are present in the case of animal cells. Nucleolus is absent and nucleolus is present in eukaryotes. Now, in the case of prokaryotes, true vacuoles are not present, but gas vacuoles are present. Whereas, in the case of eukaryotes, true vacuoles are present, but only in the plant cells. Now, apart from that, there are additional DNA present, which is known as plasmid. But in eukaryotes, it is not. So, up till now, does anyone have any doubts? Sure. All right. Now let's talk about the prokaryotic cells in depth. Now, under the term prokaryote, bacteria comes, the blue-green algae comes, mycoplasma also comes. So these three organisms majorly represent the prokaryotic cells. Now, the prokaryotic cells come in variety of shapes, variety of sizes, but remember that their cellular organization is the same. The basic cellular organization is there for every single one of them, no matter what is their shape or what is their size or anything. Now, talking about the characteristics of prokaryotic organisms. Now, the cell wall is present that is surrounding the cell membrane. So, cell membrane is the outermost layer, but even outside of that, there is another layer, a rigid layer present, which is a cell wall. The fluid matrix of the cell, meaning the cell contains the cytoplasm. Since we are talking about the prokaryotic organisms, so no well-defined nucleus will be present. So the nucleus will lack the membrane. And such a type of nucleus which does not have a membrane surrounding it, those nucleus are known as non-membrane bound nucleus, naked nucleus, right? So these are the ways by which you can describe the nucleus of the prokaryotes. Now, in the case of prokaryotes, plasmids are present, and depending upon the shape, just like how we studied in biological classification, the bacteria, they are of four types depending upon their shapes. There is coccus, meaning spherical bacteria. There is bacillus, meaning broad-shaped bacteria. There is spirillum, meaning spiral bacteria, and then there is vibrio, which is a comma-shaped bacteria. Any doubts up until now? The general characteristics is clear to everyone? Sure? What about the others? Only one student is nodding her head. And then what
Yes,
about
ma'am.
the rest of you? And you,
Yes,
Abita?
ma'am.
Yes. You switched it on before my saying. Okay. Yes, students, tell me. Sabiha, Leena, Jesslyn, Hashid, clear to you?
Yes, ma'am.
Okay. See, students, I understand that some of you can't switch on your videos because you either you don't have a device properly working or because of network issues. But despite of that, this is to every single one. Please respond in the class. When you don't respond in the class, that makes me so angry. I can't even tell you. So please don't make me angry. Am I a cool teacher or not? Tell me. You only tell me. Do you find me being a cool teacher? No response. I'm not a cool teacher. This is what you'll do. So let me be the cool teacher. I don't want to turn into a strict teacher. I really don't. Okay, I can't actually. And please don't try to make me that. So everyone respond in the class. Clear? Okay. Now, cell envelope and its modification. Now we are discussing about the cell structure of the prokaryotes, mostly bacteria. Okay. Now in this, there is a structure that is known as cell envelope. Now, if you see or hear the word cell envelope, it means that a layer surrounding the cell, isn't it? Envelope is a covering, right? So cell envelope is a three-layered structure consisting of an outer layer, a middle layer, and an inner layer. So three layers together make up the cell envelope of the prokaryotes. Now, the outermost layer of the cell envelope, one second. Okay, what is like 7 p.m. for India? Okay, dear. Like you have the test over here, Learnivio.
Yes, ma'am.
Okay. All right. So yeah. So as I was saying that the cell envelope is made up of three layers. One is the outermost layer, then comes the middle layer, and finally comes the inner layer. Now the outermost layer is known as the glycocalyx, the outermost. Then the middle layer is called as the cell wall, and the innermost layer is the plasma membrane. This is just for prokaryotes. So glycocalyx, cell wall, and plasma membrane are there. Now, this layer is chemically very complex, meaning it is made up of variety of different kinds of molecules, and it is present in most of the prokaryotes, particularly bacteria. Okay, like we can't say for a fact about the BGAs or the mycoplasma, but as far as the bacteria go, these layers are 100% present. Clear? Any doubts up until now?
No, ma'am.
Okay. Now, each of these three layers, they have their own functions to perform, meaning glycocalyx does its own function, then cell wall performs its own function, and so does the plasma membrane. But even after performing the different functions, they act together like a one unit. Okay, it is acting together like a single, and it is a protective unit. Since it is the outermost layer, it is the envelope, so it's responsible for the protection aspect as well. Now, talking about the glycocalyx, that is the outermost layer. Now, what is it made up of? So, it is made up of mucus, or it is made up of polysaccharides. Now, the glycocalyx layer may be thicker in some bacteria and thinner in some bacteria. It again varies from bacteria to bacteria. It can be thick, it can be thin also. It differs in thickness and chemical composition for different bacteria. Now, depending upon that thickness only, the glycocalyx is further of two types. The first type of glycocalyx is slime layer. Now, this slime layer is thin and it is loose and a sheath-like structure. What does it do? Since slimy layer it is, right? Slimy means sticky. So anything, if it is sticky, it will like prevent, or you can say protect the cell from the loss of water, because it will retain the water, it will hold the water. So the slime layer, it is a loose sheath-like layer, and it protects the cell from the loss of water and also the loss of nutrients. The second type of glycocalyx, depending upon the thickness, it is capsule. Now the capsule is thicker and it is a tough covering. If you compare it with the slime layer, capsule is much thicker and tougher. What does it do? It provides sticky character to the cell of the bacteria. Now, like most of you know that bacteria, like there are so many bacteria that cause diseases, isn't it? Do you people remember some of the names of bacterial diseases? Hmm?
And AIDS.
Huh?
AIDS.
No. AIDS is a viral disease.
Tuberculosis.
Tuberculosis is there. Cholera is there. Then tetanus is there. Typhoid. Yes, Jobita. Typhoid is there. Right? So these are various kinds of diseases that are caused by bacteria. And all those pathogenic bacteria, the bacteria that causes the disease, they mostly have a proper capsule present, because it helps the bacteria to stick to the body of the human being. Clear? Any doubts? Glycocalyx part is clear to everyone?
Yes, ma'am.
Okay. Now talking about the cell wall. Now the cell wall, it is a tough and rigid layer, and it is made up of peptidoglycan. Always remember that the cell wall of bacteria is made up of peptidoglycan, and the cell wall of plant is made up of plant cell wall, students.
Cellulose.
Who said it?
Cellulose.
Yes, Sabiha, cellulose. Plant cell wall is made up of cellulose. Always remember, bacterial cell wall is not made up of cellulose. Plant cell wall is made up of cellulose. Now, the cell wall of bacteria, it provides structural support, and it prevents the bacterial cell from bursting or collapsing. Since it is a very rigid layer, it is a tough layer, what does it do? It does not allow the bacterial cell to either burst, it does not allow that, or to collapse, to shrink. Clear? So this is the second layer, that is the cell wall. Now the innermost layer is the plasma membrane. Now, no matter what the organism is, the plasma membrane is a selectively permeable membrane. Selectively permeable, can someone explain what is selectively permeable?
It only allows certain substances inside the cell.
Very good. It only allows the certain substances to come inside the cell or to go from the cell to the outside. So that is what is meant by selectively permeable. Now, the plasma membrane of the prokaryotic organisms, it is very similar to the plasma membrane of eukaryotes. Almost you can say it is structurally, like in the structure, it is similar. But there are some additional things if we talk about the prokaryotic plasma membrane. Now, for example, if the outermost layer is the this thing, glycocalyx, then comes the cell wall, and then there is the plasma membrane. So if this is the cell, wait. So the outermost layer is your glycocalyx, then there is the cell wall, the second layer, meaning the middle layer. And then comes the plasma membrane. Now, what happens is that in the bacteria, sometimes the plasma membrane gets folded like this. So here it gets folded like this, and then it continues like this. Do you see the foldings of the plasma membrane? It is getting folded towards the inside, right? This is known as the extension or the invagination of the plasma membrane. And these structures that it is forming, these structures are known as the mesosomes. Clear? So the structures that are formed by the invagination or the extension of the plasma membrane are known as the mesosomes, and they are very important in the case of bacteria. Okay. Now, these mesosomes, they can be in the form of vesicle, in the form of tubes, or in the form of lamellae. Now, if I say vesicle, vesicle means in the form of circular structures, in the form of tube, so it can be present like this also, and in the form of lamellae, so it will be present in a network structure, like a proper network structure. So it can be present in either of the three forms. Clear? And now let us look at what are the functions of the mesosomes. Now this mesosome, it does the process of cell wall formation in bacteria. It does the DNA replication and distribution of that DNA to the daughter cell. It also helps in respiration, secretion. It increases the surface area of the plasma membrane as well. So these are the variety of functions that the mesosomes perform. Clear? Any doubts up until now?
Ma'am.
Okay. So this was about the cell envelope and its modification. See, mesosome are the modification that we
Ma'am,
talk.
your voice is coming from like really far away.
Oh, is it fine now?
Yes, ma'am.
See, you remember that the heading of this particular topic that we were discussing is cell envelope and its modification, right? So mesosomes are those modifications that we are talking about. From the cell envelope, particularly from the plasma membrane, the mesosome is getting formed. And this mesosome is extremely important for the bacterial cell. Any doubts up until now, students?
No, ma'am.
Okay. Now there comes another thing that is known as gram staining. Now, gram staining is a technique, and it is the technique that helps us to differentiate in between bacteria. So it is a special technique which was developed by Christian Gram. This was a person. And this technique is used to classify the bacteria into two groups, gram positive bacteria and gram negative bacteria. Now, see, what happens is that during this process, we take in the bacteria, we take the bacteria on a this thing, the slide. Okay, the slide that we put under the microscope. Have everyone here seen like used a proper microscope in your schools? Have you people used it?
Yes,
Yes.
ma'am.
Hmm. So now you know that there is a slide that is kept under the lens that you look at, right? Now what we do during the gram staining, we take that slide and we take this bacteria. Now the bacteria source that we take is curd. So we dip, like we take the curd and we smear it on the slide. And then what we do is that we add stains on it. Now, gram staining is a technique in which we add several stains, violet color stains. And then all those bacteria that take up the violet stain are known as the gram positive bacteria. And those that do not take up the stain are known as the gram negative bacteria. Okay, and in this way, all those bacteria that we have studied about, whether it is the cholera causing bacteria, tuberculosis causing bacteria, any type of bacteria, we can do the test and we can understand and we can see like whether it is a gram positive bacteria or whether it is a gram negative bacteria. And those mesosomes that we talked about, which was the modification, now these mesosomes are found in the gram positive bacteria. It's not found in the gram negative bacteria. Clear?
Yes, ma'am.
Gram staining is clear to everyone.
Yes, yes.
Why do we do gram staining? Hmm?
Clarify different bacteria to classify the bacteria to groups.
Yes, depending upon the bacteria that will get stained, those are the gram positive bacteria, and the bacteria which does not get the stain are the gram negative bacteria. Clear? Okay, now you people can note it down till here. Also, while you're noting down, you don't have to hurry, you don't have to rush. Read nicely, understand, and then only note down. Okay? Now you can switch off your video and write down comfortably. Zanab, cytoplasm is a semi-fluid like structure. We'll study about cytoplasm separately, okay? Also, students, are you done? Is everyone done?
Yes, ma'am.
Uh-huh.
Done, students?
No, ma'am.
Okay, okay, waiting.
Done, ma'am.
Everyone's done?
Yes, ma'am.
What about the others? Are you all done?
Yes, ma'am.
Yes. Yes, ma'am.
Done, students?
Yes, ma'am.
Everyone's done?
No, ma'am.
Okay, okay, waiting.
Done, ma'am. Ma'am, can you explain the capsule part again?
Sure. See, capsule is another, you can say, modification or a type of glycocalyx, wherein the glycocalyx, when it is thicker and it is tough, meaning not easy to break, then we call it as capsule. And since it is thick and it is not so easy to break, that is why it provides the bacteria the sticky character, so that once the bacteria is inside the body of the host, it will not get destroyed so easily. So that is what capsule is. Clear?
Yes, ma'am. Done, ma'am.
Everyone's done?
Uh-huh.
Done, students?
Yes, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am.
Yes, ma'am. Done.
Everyone's done? Is everyone done, students?
Yes, ma'am.
Yes, ma'am.
Done, students?
Yes, ma'am.
Yes, ma'am.
Any doubts up until now?
No, ma'am.
All right. So, yeah. So, see, if you just go through the whole entire thing, we have started to talk about the prokaryotic cell, right? Now, remember that the bacterial cell or bacteria in general, we know that bacteria can be motile or it can be non-motile. Now, students, tell me if anything, if any cell or any organism is motile, what is the structure that it has? which makes it which gives the motility or which makes it motile. What is the name of the structure?
Flagella.
Hmm?
Flagella.
Exactly. Flagella. Remember that flagella is that uh, long extension of the cell. And mostly what happens is that this flagella is actually the extension from the cell wall. It comes out of the cell wall. Okay, so if the bacteria are motile, it means that they are flagellated. So we can write about that motile bacteria are flagellated. And then these flagella are the extensions of the cell wall. Now, cell wall is which layer of the cell envelope? Outermost, middle, or innermost?
Middle.
Very good. It is the middle layer of the cell envelope. Okay, any doubts up until now? All right. You people can note this down and we'll keep it till here only for today's class. And now in the next class, we'll discuss how the flagella shape is like and we'll discuss more about the prokaryotic cell. Okay. So you can note it down and tell me when you're done.